Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Today we shall discuss about bacteriophage vector. The virus that attack bacteria is known as bacteriophage. Most of the bacteriophages infect the bacterial cell and cause its lysis. And also there is release of newly formed phage particles. Such type of bacteriophage is known as lytic phage. Other bacteriophages infect the bacterial cell but do not cause its lysis. But the genome of such phages integrate into the bacterial chromosome where it replicates along with bacterial chromosome. Such type of bacteriophage is called as lysogenic phage. Several bacteriophages are useful as cloning vector. Among them, two E. coli phages, one is Lambda phage and other is M13 phage, are more commonly used as cloning vector. When we use plasmid as a cloning vector, in that case, the plasmids are introduced into suitable bacterial cells. Then they are cloned and selected for the presence of recombinant DNA, whereas phage vectors are directly tested on bacteria alone, where each phage particle produces a plaque. The use of bacteriophage as cloning vector give us certain advantages. First is the bacteriophage vectors can accommodate large DNA fragment. Second is a large number of plaques can be screened easily as compared to the bacterial colonies for the presence of recombinant DNA. So friends, today we are going to discuss about an bacteriophage vector known as Lambda phage vector. Lambda phage vector is derived from Lambda phage, and Lambda phage is a bacteriophage, and the genome of the Lambda phage, it contains 48,502 base pair. This is the genome of Lambda phage, and the genome of Lambda phage contains 48,502 base pair. The genome has an origin of replication. It has genes for head and tail proteins. The genome also have genes for enzymes for DNA replication and also for lysis and lysogeny. The genome has single-stranded protruding end on either side. The single-stranded protruding ends are complementary and they are known as cohesive ends. The single-stranded protruding ends are responsible for circularization of the lambda genome inside E. coli. So the genome of Lambda phage is linear in phage head, but the genome becomes circular inside E. coli. And the sealed cohesive ends are known as cos site. After that, for packaging inside lambda head, the lambda DNA must be larger than 38 kilobase pair and it must be smaller than 52 kilobase pair. And we know that the genome of lambda phage is approximately 48 kilobase pair. So if we use wild lambda phage as vector, in that case it cannot accommodate larger DNA fragment. So how to solve this problem? This problem is resolved by deleting the central region which contains genes for lysogeny, and this segment is known as non-essential segment. So by deleting the non-essential segment and by joining the remaining two arms of the lambda genome, we can construct a lambda phage vector which can accommodate large DNA fragment easily. Another problem is the genome of lambda phage is so large that such that it contains more than one recognition site for virtually every restriction enzyme. So, in this case, the DNA cannot be integrated into the lambda genome. So, this problem is resolved through a process of natural selection that select only the phage particles which do not contain recognition site for specific restriction enzymes. Then you see here how the recombinant vector is constructed. This is the genome of the lambda phage. This is the cohesive end or single stranded protruding end. There is an origin of replication, and this segment, the central segment, is known as non-essential segment. The segment contains the genes which are responsible for lysogeny. And there are two recognition sites for EcoR1. So by using EcoR1, this non-essential segment can be cleaved and it can be removed by using the restriction enzyme EcoR1. So after removing this non-essential segment, we got two arms. One is left arm, other is going to be right arm. After this, the DNA insert. After this, the DNA insert, which is prepared from eukaryotic DNA by using the same restriction enzyme, that is EcoR1. This DNA insert was integrated between left arm and right arm. And this annealing between left arm, right arm and DNA insert leads to the formation of long concatemer. Long concatemer. These concatemers are appropriate precursors for packaging inside the lambda head. And lambda phage vectors can be divided into two different groups. One is insertion vector and the other is replacement vector. In insertion vector, a large part of the non-essential segment is removed. And example of insertion vector are lambda ZT10, lambda ZT11, lambda ZAP2, etc. And the insertion vector has one restriction site for a specific restriction enzyme so that DNA insert can be integrated. Other type of lambda phage vector is known as replacement vector. In replacement vector, the entire non-essential segment is removed. And example of replacement vector are lambda WS, lambda B, lambda EMBL4. This replacement vector, in this replacement vector, the non-essential segment, which is also known as stuffer fragment, is flanked by a pair of sites for EcoR1. These are the example of replacement vector. One is lambda WS, lambda B. This is a replacement vector in which the non-essential segment, which is also known as stuffer fragment, is flanked by EcoR1 recognition site. Whereas in other replacement vector, that is lambda EMBL4, the stuffer fragment is flanked by pair of restriction sites for various enzymes like EcoR1, BAM-H1 and SAL1. So friends, as compared to plasmid, the bacteriophage vectors are more useful for cloning because it can accommodate larger DNA fragments and it is very easy to screen for the presence of recombinant. Thank you friends. Please share, like and subscribe this video, and also please don't forget to give your comments. Thank you. Bye bye.